Okay, can you hear me all right? Okay. All right, so we're going to do something different this week. Give you a break, give me a break a little bit. First of all, if you did not take the quiz yesterday, I don't know what to tell you. It was due. I have not graded. I graded the ones that were turned in earlier last evening, but I didn't grade the ones that were turned in closer to the deadline. Try not to do things too late at night or I make too many mistakes. The lectures for the last several chapters are online. I'm not going to lecture over chapters 16 through 20 in class because they are posted online. So what we're going to be able to do in class then is watch some videos and have some discussions, and maybe that'll be a little more interesting, and maybe be able to do a little review that way. That does not mean you don't have to watch the videos, because they cover the content of those chapters. Now the good news is you can watch them very fast. You can speed up the YouTubes and spend less time watching them than it would take you to watch them sitting in class. So that is one advantage. Okay, I guess I better take attendance. Those of you who are watching over Zoom, I just posted a document that will go along with the video we're going to watch today. So be sure to watch, pull up that document. Anybody else here? All right. Okay. How many of you have done the lecture that is posted for chapter 16, which is the one we're doing today, on plants? At least one or two of you have. That's going to make today's discussion a little bit tougher if you haven't even looked at it. So I guess what we'll do is we will discuss that chapter tomorrow. That chapter is due today. I think it's on the schedule to be due today, correct?
Yeah.
All right. The big thing I want you to get out of chapter 16 is how plants evolved over time and what the advantages of one type of plant is over the others. What are the adaptations that help plants become so successful? And what plants are the most successful? And what I have up here on the card are examples of different kinds of plants. And we move from moss, which are the simplest land plants. Let me see if I can. I don't know if this works or not. How do I make it? Let's see. Okay, I want to be the speaker. So how do I make it so that I show up?
That's it.
Like that?
Yeah.
I don't know if this is going to show up. Is that show up at all? A little bit. Well, you all know what moss looks like, okay? The little green soft stuff, that's moss. And so that's the first category of plants that colonized the land, because plants evolved from algae, which is in the oceans. And then the second category are ferns. And what I brought in today that I was hoping to show, I think I've told you before, we have a little, when we bought our house, it has a tiny little creek and a pond. And along with that creek and a pond come a lot of ferns. So we have a lot of ferns growing in our backyard in a small area. And so this is, you know, obviously a fern. And on the back of ferns, there's little structures that are called sori. I guess you can really see them there. I was hoping they would show up better. Maybe what I'll do is take pictures of them, and then I can post those easier. But I have fern with sori on them. And you all are welcome to come up and look at these. I'll have them in the classroom the next couple days, and we'll probably have them in the lab on Wednesday. And then the third category, okay, the first category, the moss, are seedless non-vascular. That means they don't have seeds, and they do not have a vascular system. What does a vascular system do? It transports stuff, right? Our vascular system includes our circulatory system and the lymphatic system, okay? Transports stuff around. So moss doesn't have that. So that means moss has to be what? Small, because you can't transport stuff. Ferns don't have seeds. They're seedless, but they have a vascular system. So ferns can grow extraordinarily large. They're a huge fern. And then we have the seed plant. And the first category of seed plants are the gymnosperms, which actually means naked seed. And conifers, pine trees, are examples of those gymnosperms. And conifers have cones. Not all gymnosperms are conifers, but all conifers are gymnosperms. And they have different sizes of cones. And then the last category of plants are the flowering plants, which we call angiosperms. Not all flowers are big and pretty. Some flowers, like grass, very, very tiny, and often you never see them. We cut grass before it goes to seed most of the time. But some flowers are extraordinarily beautiful. Why are some flowers so pretty? What's the advantage of having a pretty flower?
It attracts the bees.
It attracts pollinators, particularly bees, other insects, and some birds, and even bats pollinate plants. So we'll look at those a little more carefully tomorrow. Okay, so make sure you do chapter 16 tonight or this afternoon. Okay, so the next thing I want to do is I gave you a handout. Before we do that, does anyone have questions, general questions about the lab report? If you do not have me for lab, you should follow the instructions of your lab professor, okay? But we're all using the same guidelines to grade your lab reports. So if you have some questions, I'll be glad to try and answer them. I sent you the data, yes? Yes. It should have an abstract, an introduction. The introduction, you may want to write this, well, you guys have access to the recording, but it needs an abstract. An abstract is like a summary. It does not have any citations in it. What it generally contains is a sentence or two from each of the other sections. It'll have a sentence or two from the introduction, a sentence or two from the materials and methods, a sentence or two from the results, and a sentence or two from the discussion. Okay? It should include the result that says this is what happened, and then in parentheses you would have the statistics. T equals P, degrees of freedom equals P equals or P less than, or whatever you can do that either way. Okay? Everybody understand that? That should be in the abstract. The title page. I actually am picky about how you do the title page. There is an instruction in the guidelines that shows you a picture and gives you a description of what the title page should look like. It's 4 inches from the top. I think it's bold and all capitals for the title. And then for your name and class and date are normal type and single space. So you got to do these things correctly. Okay? And they're centered on the page. And then you start the next page with the abstract. You do not do a running heading. A lot of you on your short lab reports put some sort of heading on each page. We don't do that. The only thing that should be starting with the abstract is a page number at the top right-hand corner. Your name should not be there. Just 1, 2, 3, 4, however many pages it is. Okay? You do not have to start a new page for a new section, but if the new section, what is the general rule? I think if you're only going to have a couple lines, you're supposed to go ahead to the top of the next page. So don't leave just the heading at the bottom of the page. You double space between sections. That means you hit return for double space twice. The section headings are centered and they're bold, all caps. Then normal double space. Then you write your whatever it is you're going to say, and then when you get to the next section, you double space. You put the next heading. You do 1 double space, and then you write whatever that section says. Okay? The introduction should include at least 1 primary source, and we provided you a couple of primary sources, so you don't even have to go searching for primary sources. Okay. Was the first reading a primary source? No. Then the materials and methods, you say what you did. For the results, should include a graph. How many bars should be on your graph? 2 bars. What should the 2 bars be? How close you could get to the squirrels, right? For the cemetery and for the campus. Okay? Make sure your scale is not too big. Make sure your scale is reasonable for what the data is. Let's see. You should have a couple of sentences writing with the results. And generally your last sentence in the results is the sentence that says, this was statistically significant, here's the statistics. Then the discussion, and this is the place where people usually do the worst. Okay, so make sure you read the section on discussion. Discussion is not a repeat of the results. Crap. People online, the electricity just flashed. You probably can't hear me. The new message I sent, what?
If you're gonna have to do.
Oh, I forgot to put on the, they can use the old, actually, they can use the old invitation, it'll still work. There's Ashton back. Okay. We will try this again.
Wait, did you hit record?
No.
It's on, I still don't like
Okay, you've got the beginning of it though, right? Okay. All right. So what were we talking about? What were we talking about?
it. The discussion.
The discussion. Okay, so what should be in the discussion?
The interpretation of the data.
The interpretation of your data. So this is where you might bring in some information from your source, something about squirrels and why they tend to distance from people or something like that, and then bring that in talking about what your results were. You might talk something else about how people on campus feed squirrels. You know, you might do something that would explain your results, but you don't just give the results again. The last sentence usually of any discussion refers to what further or additional research should be done. You always refer to what would be the next step. What would be the next thing we would do? Further research would be necessary to determine yada, yada, yada. Okay, so something along those lines is what you would do to figure that out. I don't know how to get rid of that. Okay? Then the last section is your literature cited. It is not called references. It is not called sources. It is called literature cited. The format for literature cited is different than the rest of the paper. You have to look at the guidelines to see how you're supposed to cite your source. For one thing, it's single-spaced. For another thing, it's outdented instead of indented. What does that mean?
Hanging in
It means the first line is all the way to the margin and the second line is indented. That's called an outdent. I don't know why, but that's what it's called. Okay? And it's single-spaced. And you have to follow the format that it tells you to use in your guidelines. When you don't do the formatting correctly, you are just costing yourself points for no good reason. Okay? Here's why I'm picky about it. When I turned in my dissertation, we had to have it printed on 100% cotton paper. Well, 100% cotton paper costs a lot of money. This is before digital everything. And it had to be that way because it had to be preserved. So you had to turn into the graduate school 2 copies of your dissertation on 100% cotton paper. And so I had to go to Kinko's and get it copied, and it cost a whole bunch of money. I had a baby. Let's see, this would have been in October, November. He was born in March. This is before there were car seats that had little handles on them. I had to physically carry him or have him in a stroller or something, okay? And I trot up to, I've already defended my dissertation. Everything's over. I just have to turn everything in. And this woman, who's like a secretary, is sitting at a desk. You know, there's like a window. I take it up and hand her the little box that has the dissertation in it. And the first thing she does is opens her drawer and pulls out a ruler. Thought I was going to pass out. Because what she did was measure the margins. Okay? And if you did not follow the instructions, they would reject your dissertation. Which means I had to retype it, recopy it, and turn it back in. And I was sweating bullets, let me tell you. Then she tells me I didn't print it properly. Because back then, using a laser printer today is really not that big a deal. But back then, there weren't inkjet printers. There were what we call dot matrix printers. Those dot matrix printers are the kind that do little bitty dots and to make up your letters and numbers and stuff. And that's the kind that has the little edges on it that you pull off. You know what I'm talking about? They still have them like in the registrar's office where they're going to print out whole bunches of documents, and a lot of times they're two or three copies of documents. And so instead of just throwing ink on a page, you're actually hitting it with something. Okay, so that's what I printed my dissertation on, and it was called. You had to use what's called near letter quality, and I used near letter quality. And she looked at it and she said, "I don't know if this is going to pass or not." I mean, I was dying because here's the secretary who has the power to tell me I'm not going to get my degree because I didn't follow the instructions properly. Now, I have a great deal of respect for secretaries, especially after this incident. Always treat secretaries really, really nice because they have power. You should treat everybody nice. Treat secretaries and waitresses and people nice, okay? She took it, but I got a very stern talking to in the process. So that's the reason that I am very picky, because if you submit an article to a journal and you don't follow their formatting directions, they will kick it right back to you, and it's just too bad. It could be the best research in the world, but if you're not going to follow the instructions, they're not going to publish it. So don't lose points on a lab report because you don't follow the directions. Okay? Just do what it tells you to do. You should have a printed out copy of the guidelines. If you don't, there's bunches of them down in the lab. It's a packet that's about this thick. It's also on Canvas, as is a separate copy of the rubric that we're going to use to grade the lab reports. Okay? So does anybody have a question about the lab reports? They are due at midnight on Wednesday. At least my class is. I'm not sure what time the other lab instructors put, but mine are due midnight on Wednesday. Okay? Any other questions about the lab report? Everybody comfortable with what's expected of them? You have the data. Hopefully you have it if you're in another class. Okay. All right. Anything else you have questions about? I think our exam, if I'm not mistaken, is next Tuesday morning. Is that right? I think it's from 9 to 11. No, Wednesday. Maybe it's Wednesday. I think it's Wednesday. I'll have to look it up. Actually, I've probably got it on my calendar. I do not have it on my calendar. I think it's next Wednesday, 9 to 11, yes.
How do we see our whole final exam schedule?
I'm sorry.
How do we see or find our whole final exam schedule?
Well, let me see if I can find it.
For every class?
Yes, there's one for all classes. What I generally do is go to registrar, I go to Georgetown College, registrar, final exam schedule. that's the wrong year. I know I've seen a copy of it. Oh, no, that's fall of 2019. Okay, that'd be good. I'll have to see if I can find, I probably got an email that has it, and I couldn't tell you where that email is. I will have to go find it, but I'm pretty sure, let me try one other thing. If I go to my email, did you find it?
No, that's not the question. Is our final gonna be over all the chapters or just like 16 through 20?
It will be all the chapters.
Okay.
I don't know. I can't find it right this second. So I will have to look that up.
If you ask your advisor, I think they post the link, like each individual advisor, like I know my.
Well, I'm an advisor, so I should.
Like the academic advisors, I know for ours that they gave us a link.
Yeah, if you're in foundations, I would think, or freshman seminar, they probably.
Seminar, that's who told us.
Your freshman seminar professor probably has it handy, but evidently I don't have it right handy, but I'm pretty sure that ours is on Wednesday from 9 to 11, I think. All right. Did you say somebody have another question? Oh, it'll just be use the study guide for each exam. We will have an exam, there should be another exam scheduled, right? Well, there's gonna be another exam, it'll probably be Friday.
Is that like 16 through 20?
It'll be over, yeah, over this material. The more exams you have, the less each one counts. You guys need to understand that.
You want to add a third? You want to add a third?
If I don't give you another exam, I can't replace the lowest exam with the final exam score.
Okay, I'm fine with that one.
Yeah, what if we just end up doing vertical final days and we did all other debates as well?
Then I don't replace it.
After 16 million years on Earth, the rhinoceros.
Okay. Now, what I need to be able to do here is share the screen. Take this away. Okay, this is the note-taking guide for this video. I hope I gave you the right one. I will tell you up front, I'll tell you when we're switching from part 1 to part 2. It doesn't like tell you when it's switching. The first 3 sections are kind of depressing. I do not show this to make you depressed. That is not the purpose. It's got some really interesting information in it, and then the 4th one is a little more upbeat. And then we'll watch another video that has some really gorgeous photography in it. So hopefully you'll get something out of these.
Are we turning the papers in?
What? You are not turning these in. These are just note-taking guides.
Okay.
My children tell me I should not give you these because all you pay attention to then is what the question is, answering the questions on the sheet. Okay? I don't know.
Are these notes or anything going to be on test?
Anything that's in these videos, fair game for the test.
Oh.
Whether there's a question on it or not. Okay? So pay
Is
attention.
on the
Uh-oh.
verge. After 60 million years on Earth, the rhinoceros is on the verge of extinction. Soon, elephants may exist only in zoos or protected areas. Half of all primate species are now rare or endangered. Despite heroic efforts, many of the world's wild creatures are doomed. The loss of species is now the same as when the great dinosaurs became extinct. Will these creatures become the dinosaurs of our time? Today, mankind is changing the entire face of the planet Earth. We are replacing the natural world with our own. We are destroying our environment at a frightening rate. We are tampering with the future course of evolution. And still we are amazed by the diversity of life. Of the millions of Earth's creatures, no species is alone. Each is linked to the life that surrounds it. Each holds a unique place in its environment. We are learning that the only way to save species is to save the places where they live. But today, much of the living world is poised on the threshold of extinction. On this fragile planet, life itself may hang in the balance. Because there is always knowledge to be gained, new paths to explore, and mysteries to unlock, the infinite voyage is made possible by a grant from Digital Equipment Corporation.
Okay, this is the beginning of part 1.
This barren valley near Pueblo, Colorado, is an intriguing chapter in the history of life on Earth. 100 million years ago, this was the bottom of a vast inland sea. Today, it is one of the richest graveyards in the world. Geologist Earl Kaufman has come here to find out what happens when life becomes extinct. He is interested in a cataclysmic event that once destroyed 2/3 of all living things in the sea. The death of life is common throughout Earth history. Layer by layer, the story unfolds of how long ago life went over the edge.
Clear this slope off here and then put fresh rock and be fresh rock for kind of the same thing.
Kaufman reads the layers like the pages of a book.
How dramatic that color change is. That's the beginning of this ocean chemistry that we see. Below the color change right there in the rock. That'll be our reference point. Okay, so the second nail will then go 10 centimeters higher, right at color change right at that point. And that will define our first sampling interval. Okey-doke. Should do that this way.
To find out what happened to the creatures that lived here, he takes one sample every 1,000 years, before, during, and after the extinction.
So this is the pre-extinction level, right?
Sample number 18.
Sample 18 centimeters.
In his laboratory at the University of Colorado, Kaufman counts the number of fossils in each sample.
Then we begin to pick up a few more fossils here. As you can see, we have those. Oh, yeah, Dinoceras again.
That's a good healthy bottom fauna. There are also juveniles of the same species. And we have an ammonite, Stomohamides, in here.
In the early samples, Kaufman would identify 350 species, but the next sample shows no signs of life.
We'll avoid the dead zone.
Yeah, we should be in it. Definitely, there's not one fossil on those surfaces there, so the extinction is virtually complete. And there's no disturbance whatsoever. No burrowing, no nothing. The bottom is dead. This is interesting because here's one fossil, one species. Looks very small, probably a plant of some sort. But otherwise, it's not a fossil of rock and totally undisturbed.
At first, species gradually declined, then suddenly, mysteriously, they disappeared. Kaufman now believes the fate of one species may be closely linked to the fate of another.
Species that comprise an ecosystem are not just separate species, but rather they're closely linked ecologically into a type of a structure, into food webs, into symbioses or obligate relationships between organisms. And considering this then, a mass extinction is not just the loss of one species at a time, but the loss of species until some key species in the food chain or some key species or set of species in the ecological structure are affected. And when they go, the entire ecosystem will collapse around them very rapidly.
To look for the key species, Kaufman samples are examined for other organisms.
This
The
looks
first link
like a
in the
nightmare to
oceanic
me.
food chain were creatures smaller than a grain of sand, called plankton.
Can you imagine if that was
The
your
plankton
job?
are painstakingly separated and classified. Before the extinction, there were some 60 species. Then their numbers began to decline. When the plankton fell below a critical level, larger organisms began to disappear. With the food chain broken, the system collapsed. The balance was tipped, and life tumbled into extinction. Could this ancient pattern occur again? Earl Kaufman.
Our research shows us that mass extinctions are relatively common in the ancient record. They occur every 26 to 30 million years, roughly cyclic nature, perhaps triggered by extraterrestrial phenomena like comet and meteorite impacts. If that cyclicity holds, then we can expect the next natural mass extinction to occur 12 to 14 million years from now. But on the other hand, if we look at the modern world, we see that the rate of species and diversity loss and the destruction of habitats that is going on today parallels in magnitude virtually all ancient mass extinctions that we know well. Except this mass extinction is not triggered by some extraterrestrial phenomena and not even natural changes in the global environment. It's triggered by man, man's destruction of habitat, man's destruction of the diversity of life on Earth, man's pollution of the atmosphere. Man, in fact, may be regarded as a human meteorite, but one that is unpredictable in cyclic mass extinction theory.
Today, man has the technology to change the world. Nature is disappearing, replaced with crops, stripped away from resources, paved over as cities expand. Human need and human waste are threatening the web of life. In 40 years, the human population will double from 5 to 10 billion. But already our species is overrunning the planet.
They actually do that in Japan.
A tapestry of lights trap the human presence across the earth. The Sea of Japan is ablaze with the lights of a gigantic fishing fleet. Gas fires in the Middle East outline the oil-rich Persian Gulf. Fires in Africa fuel the struggle against famine in the Third World. Forest fires in South America signal the end of an untouched wilderness. Less than 100 years ago, no city lights were visible from space. Now they glow like stars against the night sky. With the coming of the space age, our view of Earth took an epic turn. For the first time, we saw a blue oasis, fragile and delicate.
We're still in part one.
Now, with satellite technology, we can monitor the health of planet Earth. At NASA's Goddard Space Flight Center in Maryland, Compton Tucker draws from a vast library of satellite images to construct a unique map. This is a map of life. It follows changes on the planet from one season to another. Over the past 8 years, Tucker has observed subtle shifts in the Earth's vegetation.
Discovery, we're looking down at Houston. Looks like they're real clear down here today. It's a pretty view.
October 1988, the shuttle Discovery returns to space on a mission to photograph the Earth.
Our environment is a very fragile thing. I think that's something that we're very impressed by when we take photos and as we look out the windows.
As the astronauts fly across the Amazon, they are stunned by what they see.
When you fly over South America, you see these incredibly large fires. It must be 10 miles across, all over, especially the Amazon basin, where they're burning off the vegetation and it just fills the whole continent with smoke.
Compton Tucker has been monitoring these fires since 1982. His images confirm that in one state of Brazil called Rondônia, the forest is being destroyed twice as fast as previously thought. Here, 14,000 square miles have been burned in the last decade. The destruction was triggered when a single highway was cut through the forest. Now a network of roads, visible from space, has grown like the branches of a geometric tree. In Rondonia, the opening of the forest frontier echoes the settlement of the American West. But with modern tools, homesteaders are transforming the wilderness with deadly efficiency. An ancient rainforest is becoming fields and pastures. If the trend continues, by the turn of the century, 55,000 square miles of Rondonia will have gone up in smoke. For many years, environmental scientist Norman Myers has voiced his concern about the destruction of rainforests.
I have some images here for the Amazon Basin, Rondonia, Santa Rosa, and Acre.
Compton Tucker has invited Myers to review his most recent findings. One image is particularly disturbing.
Quite a few fires there. So 2,500.
Yes, now that's just an estimate, say 2,500, perhaps plus or minus 300 or 400. Now, each point we see is 1 kilometer by 1 kilometer, and it could be within that area. We could have 2 or 3 smaller fires burning.
Sure.
So that's why we say approximately that there are at least 2,500 fires actively burning at this particular time, about 2:45 in the afternoon, on the 9th of September of last year.
This area, now just outside of Montana, those fires are causing the extinction of at least 1 plant species per week, and maybe 2 or 3, maybe more animal species, mainly insects, per day. If NASA, with one of its space probes to Mars or somewhere, found even a single life form on one of those planets, it would make headline news. There'd be more probes sent off to have a look at this little thing, this extraordinary phenomenon. And yet here we are, evidence on this screen that we're pushing species down the tubes on our own planet at a rate of several per week. It seems so ironic.
Extinction rates are imprecise, but Myers is alarmed by the trend.
Now, some people might say, well, if we lost several species yesterday, but the sound still came out this morning, and the newspapers came out on time and so on, does it really matter? It does matter in the long run. It matters enormously, absolutely. The best way to think about it is to Paul Ehrlich's comparison of the aeroplane. Pretend you're getting on an aeroplane, and you see a mechanic taking rivets out of the wing. And you say to him, what are you doing that for? And he says, well, we need them for another aeroplane. And it doesn't matter because every aeroplane has tens of thousands of rivets, so a few won't matter. Would you really get on that aeroplane? Wouldn't you say to yourself, how do we know when a few becomes many, becomes too many?
Okay, I want to make sure you understood. It's a little hard to understand. What is a rivet? Okay, he's talking about an airplane. What is a rivet? Anybody know what a rivet is? What's a rivet? It's a fastener. It's basically like a big bolt, okay? A huge big bolt, okay? Your jeans might have rivets in them, okay? Little bitty rivets, all right? It's kind of a metallic thing that holds 2 or 3 things together, okay? So his comparison, or Paul Ehrlich, he's using Paul Ehrlich, who's a very famous scientist, example is if you're getting on an airplane and you see a mechanic and he's removing rivets from the airplane, that means he's taking out bolts. And you say, why are you doing that? And he says, well, I need them for another airplane. And besides, the airplane has hundreds of thousands of rivets, so it won't matter if I take out a few. The question is, would you get on the airplane? How do we know when you've taken out too many? And what is the analogy of the airplane compared to losing species? So how do we make that connection? Okay, so each bolt represents a species, and so if you remove species, then what? What? Once they've gone extinct, so what does that matter to the environment? Makes the environment more unstable, because we honestly don't know until after the fact which ones were the most important ones. Which ones, when you remove them from an environment, can cause the collapse of the environment. We may not know. We have better tools to predict today than we did 100 years ago, but we still don't know with certainty if you take out this species, is this going to be a problem? So that's the comparison.
What splendid creatures once lived here, we will never know. But one fact is known: there is no return from extinction.
Okay, let's talk about one other thing. Why are they burning the rainforest? Yeah, to make more space for pasture, to build factories, to do all that kind of stuff. Why is that a problem? Didn't we do that? We meaning the United States of America, didn't we do that?
Brazilian rainforest is like a big place that takes in a lot of our greenhouse gases. So it's more important to have that in the world supply.
Okay, one problem is that rainforests are so productive in terms of taking out the CO2 from the atmosphere. But why do they want to do this? What did we, meaning people in the United States, what did we gain by cutting down our forests? We didn't cut them all down, but by cutting down the ones we did cut down. What was the gain? What was the value? Why did we do it? We got more what? We got more land for agriculture.
Is that more powerful?
Became more powerful. We built an industrial. We went from being, humans went from being hunter-gatherers to being agrarian, which is farming, to now being more industrial, and we're considered now maybe post-industrial, whatever that's going to be, digital, I guess, next. And so in the process of making farmland, I used to think farmland was just like, it's gorgeous, it's pretty, but it's really very destructive to the environment. That's not natural, okay? And then you build industry. Why does Brazil want to do this? Why do they want to why do they want to burn down the rainforest and build agriculture and build? To get more money. Why do they want more money? They're not a rich country. Brazil's not too bad off, but they want what we have. Let's be honest. And do you think they're happy when we go down there and say, please don't cut down your rainforest? It's taking out the CO2 from the atmosphere. Do you think they care? No. They want to feed their people. Let's give them the best possible motives. They want to feed their people. They want their people to have education. They want their people to have jobs. They want their people to have opportunities. They want their people to have stuff. Same as so-called progressive Western world. Okay? What we say is, but rainforests are different, and they are different. Here's the problem with rainforests. When you cut down a temperate forest, a pine forest or an oak maple forest or something like that, and then you abandon it, it regrows. Kentucky used to be, when the native peoples were in charge of the continent, it was the hunting ground, and they burned the forest back then to control it, but they didn't live here as much. And if we abandon, if people completely abandon the state of Kentucky, eventually the trees and stuff would grow back because the soil is fertile. In a rainforest, the soil is not fertile. And what happens when you cut down a rainforest is it won't support agriculture for very long because the soil is not good. And you can grow crops on it for a few years and that's it. And then you have to cut down more rainforest. And the rainforest won't grow back because it takes the whole ecosystem to keep it going. And in fact, when you cut down, by definition, a rainforest gets lots of rain. You cut down a rainforest, you basically end up with a desert because you don't have the cooling effect of all the vegetation and stuff, and so it becomes hot and dry, and then it doesn't dump rain down it because it doesn't cool down the moisture and turn it into rain. So you go from being a rainforest, which is a very productive ecosystem, to being a desert, which is a very unproductive ecosystem. So that's the problem. And so the people in areas where there's lots of rainforests are not too thrilled when the big bad American comes in and says, please don't cut down your rainforest. Right? All right, so we're starting part 2.
The Manu River in eastern Peru, a last great unspoiled wilderness. A pioneering group of Peruvian and American biologists, led by Terry Irwin of the Smithsonian Institution, has accepted an extraordinary challenge. They are here to inventory one of the richest concentrations of life on Earth. The project is called BioLab. For the next 4 weeks, a remote ranger station will be home. From this base, the group will identify and catalog every plant and animal they can find. Their goal is to construct a detailed anatomy of a threatened ecosystem before it disappears.
We're not talking about just the extinction of a few species. We're talking about, under the present mode of deforestation, we're talking about extinction of the evolutionary process. This process that here around us and in this surroundings has taken 40 million years to develop. BioLab wants to address very quickly the inventory of species, how these species fit together, so that we can make predictions on how to save this wonderful biological diversity for the future.
Three-quarters of all living things can be found in rainforests, most of them high in the treetops. The larger species are more familiar. They have long been exotic symbols of the Amazon jungle. These creatures represent only a fraction of forest life. Their existence depends on a less visible world, a world even more mysterious and complex. dazzling array of plants and insects in a rainforest is testament to the creativity of life. Each species has evolved a unique strategy for survival. Katydids outwit their predators by mimicking leaves, one dead, one alive. Toxic chemicals in the spines of a caterpillar teach birds to leave it alone. Plants also use chemicals for protection. But a highly organized community of leafcutter ants can strip a tree in a matter of hours. Each creature, in his own way, has found a home in the forest.
Look at those bananas.
Look at how big that is.
Spines on the thorax. The needle spire that can stay on the edge of the tree. Very close to. Okay, back to work.
To undertake the study of this fascinating world, Terry Irwin has devised a clever plan. The forest is divided into 100-yard plots. Prominent trees are identified and labeled. Their exact locations are entered into a computer and will become reference points within the plot. Maps are then generated to guide researchers in the field.
That's a dot matrix printer.
Botanist Robin Burnham samples leaves from the forest floor.
It is quite a bit. You see here, other ones that I've collected from the same forest, same type of forest, include as many as 50 species in a single half-meter square quadrant. That's rather surprising to me that we can get 50 species of leaves in an area that small, because if you think of it, surrounding an area this small with 50 different species of trees is going to be pretty tight, and that's probably one of the indications of the high diversity of forest that I'm looking at.
Grace Servat, a Peruvian ornithologist, stretches nets across the corridors of the forest so low-flying birds can be caught and identified. Piranhas inhabit the Manu. Still, biologists sprayed its backwaters.
Did you catch that piranha?
The river teems with life, but even in the tropics, a fish is hard to catch.
Oh, it's unfortunate.
At sunset, the forest comes alive with creatures of the night. Bats play a critical role in the life of the forest by dispersing seeds, and Don Wilson is concerned about their future.
Bats all over the world are having problems because of forest destruction, loss of habitat. Places like this probably have a greater diversity of bats than just about anywhere in the world. Get a good look at his feet there. He uses those feet to scoop insects off of the surface of the water, probably.
That's a good one. And the second one tonight? Oh, excellent. That's the first one of those I've seen on the trip. Where did it go?
Oliver Flint has been collecting insects since his early teens. After 45 years, he's still excited by something new.
So when I see something that looks new and different, not quite so common, I'll make a special grab for that one. If I can survive with all the stuff flying in my face all the time.
One sort of case that
not really easy.
Terry Irwin prepares to explore life in the treetops. He has designed a systematic way to investigate.
Okay, we'll stop there. Pick it up tomorrow. Okay, so for tonight, you need to make sure that you have finished chapter 16. And then tomorrow you need to do chapter 17, and then there's, I combined 18, 19, and 20 into one longer lecture because a lot of material overlaps. And these videos overlap diversity, which is 16 and 17, with ecology, which is 18, 19, and 20. So that's why we're kind of looking at it all together. Okay? I don't have any way to do that. She asked if they're going to be on, they're not available online. They're kind of old. I can give you I can post links to parts of them, but it won't be like the whole thing. If you're going to be on campus, I can loan them out at some point, or you can come watch them at another time. It would be the best thing to do.
The exam's on Friday?
It may be over the week. It may be Monday. Maybe Monday.